Picture this. You're looking at a mother mouse. Normally she's peaceful, maybe a little timid, just kind of minding her own business.
Yeah.
But the second she gives birth, something invisible flips.
Right. It's like a total transformation.
Exactly. An intruder gets a little too close to her newborns,
Yeah.
and instantly she transforms. The timid mouse becomes this, fierce, relentless protector. It is the ultimate universal mama bear instinct.
Oh, absolutely.
And then, just as quickly, when the pups are fully grown and leave the nest, that fierce protector vanishes. She goes right back to being peaceful.
It's a behavioral switch that is so drastic and so perfectly timed that it almost looks engineered. I mean, it's practically binary, on and then off.
Which is exactly what we're going to get into today, because that's our mission for this deep dive. We are going to decode the exact biological algorithm, the specific neural hardware, and the chemical software that allows maternal aggression to rapidly turn on after birth and, switch off the second those babies are gone.
It's a fascinating topic.
It really is. And we are pulling from a landmark Nature paper by Yamaguchi and colleagues. We're going to examine all the details, the microscopic mechanisms, and the actual brain rewiring that makes motherhood possible.
And to understand why this specific paper matters so much, you really have to look at the historical context. For nearly 50 years, science has kind of, known the basic recipe.
Right, like the broad stroke.
Exactly. We knew that pregnancy hormones, combined with the physical act of pup suckling, somehow caused this aggressive behavior, but it was a black box.
We couldn't see inside.
Yeah, we knew the inputs and we saw the output, but the precise microscopic brain rewiring, the actual microcircuitry making it happen, has been a complete mystery until this research.
So to crack open that black box, we need to start with the hardware, the physical brain regions that make up this aggression circuit. There are two main areas we're looking at, right?
Yes, there are two specific populations of cells we really need to map out. First, we have a region called the PA,
Which stands for?
the posterior amygdala. And specifically, the researchers isolated cells in this region that express estrogen receptor alpha. So in the paper, they refer to them as PAESR1 cells.
Okay, posterior amygdala. And the ESR1 just designates the estrogen receptor aspect.
Yeah.
What is the actual function of these cells in the context of the mother mouse?
Think of the PASR1 cells as the brain's threat detection radar.
The threat radar. Okay, I like that.
Yeah, it's the sensory relay that identifies a potential danger in the environment, like an intruder approaching the nest.
Got it. The threat radar. And this radar obviously has to communicate with something to actually initiate a fight.
Precisely. The radar sends a signal to our second region, which is the VMHvl.
That's a mouthful.
It is. That's the ventrolateral part of the ventromedial hypothalamus. And inside the VMHvl, the researchers zeroed in on cells expressing neuropeptide Y receptor 2.
So they call these VMHvl and PY2R cells.
You got it.
So if the key A is our threat radar detecting the intruder, the VMHvl is like the action command center.
That's a great way to look at it. When these VMHvl and PY2R cells fire, they basically pull the behavioral trigger that initiates a physical attack.
Okay, so the pathway is the threat radar sending a signal to the action command center. But, hypothesizing a pathway is one thing. How did the researchers actually prove this belief connection is the hardware for maternal aggression?
Well, they used a turn-and-off experiment relying on this fascinating technique called chemogenetics.
Yeah.
They took a specific breed of mice, C57, and Swiss Webster F1 hybrids.
Why those hybrids specifically?
They chose them because they display highly robust, reliable maternal aggression, making them perfect for this study. So using a virus, they inserted a designer receptor into this specific PA to VMHvl pathway. This receptor is called HM4DI.
Okay, and what does this HM4DI receptor actually do to the neurons? Does it change them right away?
On its own, absolutely nothing. That's the beauty of a designer receptor. It just sits there completely inert until you introduce a specific chemical key that doesn't naturally exist in the animal's body. In this case, the researchers injected a chemical called CNO, or clozapine oxide.
Let me see if I'm tracking. They basically built a custom lock on the cells and then introduced the custom key.
Right. And when CNO hits the HM4DI receptors, it acts like a remote control kill switch. It instantly silences those specific neurons.
So what happened to the mother mice when the radar was silenced? Did they just shut down?
No, they completely stopped attacking intruders. The maternal aggression dropped to 0. But they didn't become catatonic.
Oh, really?
Yeah, they still investigated the intruder, and they still cared for their own pups perfectly. Only the aggression was turned off.
That's a crucial distinction. It proves this specific pathway isn't responsible for, like general maternal care or just basic social interaction.
Yeah.
It is a highly specialized, dedicated circuit just for fighting.
Exactly.
So that's how they turned it off. Did they also test turning it on?
They did, using optogenetics. So instead of chemicals, this technique uses light. The researchers introduced a light-sensitive protein called channel rhodopsin 2, or CHR2, into the pathway.
Light sensitive. Yeah. They then implanted extremely fine optic fibers directly into the brain. Optogenetics allows for incredibly precise split-second control.
Wait, how exactly does light make a neuron fire?
The moment the researchers pulse a very specific wavelength of light, in this case, blue light, at exactly 470 nanometers, the CHR2 proteins physically open up.
Oh, I see.
Yeah, positively charged ions rush into the neuron, and it fires an electrical signal. They are manually forcing the threat radar to talk to the action center.
And when they flash that 470 nanometer blue laser in the lactating mothers?
Instant time-locked attacks.
Yeah.
They essentially remote-controlled the aggression.
Hang on, I'm a little confused by something in the study design here. If they mapped this physical pathway, and they have a laser that forces the pathway to fire, did they try this on female mice that didn't have pups?
Yes, they did. They took virgin female mice, identified the exact same pathway, introduced the exact same light-sensitive proteins, and fired the exact same blue laser.
And did they attack?
Nothing happened. No attack whatsoever. Wait, why? If the physical hardware, the threat radar, and the action center is present in both sets of mice, why did only the mothers attack when the laser forced the cells to fire?
That is the pivotal question that drove the next phase of the research. It turns out, while the basic brain regions are the same, the functional hardware isn't exactly the same. The physical connections in the brain have fundamentally transformed due to motherhood.
So let me do a quick recap here. We've established that the PAE's R1 to VMHvlM PAPR2R pathway is the necessary and sufficient hardware for maternal aggression.
Oh.
But it's clearly different in mothers. So think about this for a second. Think about a time you felt instantly defensive over someone you care about. Could you feel your own threat radar talking to your action center? Because what we're about to see is that this isn't just about hormones floating around. We're talking about a physical, structural upgrade to the brain.
Exactly. The researchers looked at synaptic plasticity, how the connections change, and excitability. To measure this, they used something called in vitro patch clamp recordings.
I've heard of patch clamping, but how does it actually work in practice? It sounds complicated.
It is one of the most delicate techniques in neuroscience. You take a microscopic glass pipette and literally seal it against the membrane of a single individual neuron in a slice of brain tissue.
Wow.
Yeah, this allows you to measure the exact electrical currents flowing in and out of that one specific cell.
So they are essentially wiretapping a single neuron to see how much signal it's receiving. Let's use a visual to help picture this. Imagine the connection between the threat radar and the action center as a physical bridge. How much traffic is actually getting across that bridge?
To measure that traffic, they looked at OEPSCs,
Which means?
optogenetically evoked excitatory postsynaptic currents. They flashed the optogenetic light at the radar end of the bridge and used the patch clamp to measure how much signal actually made it across to the action center. And the difference between virgins and mothers was staggering.
What were the numbers?
In virgin mice who were in estrus, only 12% of the action center cells received any signal at all from the threat radar.
12%.
Yeah. The connection was incredibly weak. Almost no traffic was getting across.
And what happens to that bridge during lactation?
The connectivity explodes.
Yeah.
In lactating mothers, nearly 50% of the cells show direct spiking, and a massive 79% show those excitatory currents, the OEPSCs.
That's not just a minor adjustment. That's going from a shaky, narrow footbridge to a massive, multi-lane concrete highway. The physical connections became wider and significantly more numerous in just a matter of days.
But the upgrade doesn't stop at the synapses, the connections between the cells. The action center cells themselves, the VMHvl and P2R neurons, change their intrinsic excitability.
You mean they change how they process the signal once it arrives?
Right. The researchers measured two specific properties. First, the rheobase.
Rheobase.
Yeah, which is the absolute minimum electrical current needed to make a neuron fire a signal. Second, they measured the RMP, the resting membrane potential. Interestingly, the rheobase and the RMP didn't actually change between virgins and mothers.
So if we think of the action center as a car engine, the resting membrane potential and the rheobase are like the idling speed and the minimum amount of gas you need to press to get the car moving. And you're saying that minimum threshold stays the exact same.
Yes. It takes the same minimum amount of juice to start the engine. But what did change drastically was the maximal firing rate.
Oh.
Once that minimum threshold was crossed, the cells in the lactating mothers fired significantly more spikes much faster than the cells in the virgin mice.
Okay, so expanding our analogy, not only are there more lanes on the highway bringing signals to the action center, but the action center itself has been upgraded. It went from a regular commuter engine to a high-revving race car engine. It is highly sensitive and capable of a massive output.
And if you put those two things together, the strengthened synaptic bridges and the hyperexcitable action center, it entirely explains why your virgin mice didn't attack when the laser fired. Their bridges were too narrow, and their engines weren't revved up. The light couldn't push enough current through to trigger the aggressive behavior.
So, just to summarize this second major piece, lactation physically rewires the brain by increasing the number of active connections from the PA and boosting the firing capacity of VMHvl and IPPR cells. It makes you wonder, if your brain can physically upgrade its hardware in just a few days to handle a new life phase, what other latent circuits might you have waiting to be unlocked?
It's a profound thought.
But wait, this brings up a huge logical dilemma.
What's the contradiction?
Well, the VMHvl, our Action Command Center, is famous in neuroscience because it's heavily involved in controlling both fighting and mating in female mice.
True.
So if this threat radar, the PA, is suddenly blasting all these massive excitatory signals across that new multi-lane highway into the VMHvl, why don't the mother mice get completely confused?
Yeah.
An intruder shows up, the radar fires. Why don't they initiate sexual behavior instead of fighting?
It's a brilliant question because the potential for a biological crossed wire is huge here. The answer comes down to cellular specificity. The VMHvl isn't just one big uniform blob of identical cells. It's highly compartmentalized. We already know the MPI2R cells handle fighting, but there are entirely different subpopulations of cells right next door.
Living in the exact same neighborhood.
Exactly. Specifically, there are cells expressing shakar and another group that are NPI2R negative but ESR1 positive. These specific populations mediate female sexual receptivity, a behavior known as lordosis.
So you literally have fighting cells and mating cells clustered right next to each other. How does the threat radar know who to talk to when it builds this new highway?
The precision of this brain plasticity is astounding. The researchers used another optogenetic tool called Chronos GFP.
Chronos GFP?
Yeah, this allowed them to map the exact pathways with incredible resolution. They found that during lactation, the PA doesn't just indiscriminately blast signals into the entire VMHvl neighborhood, it selectively strengthens its connections only to the fighting cells.
And it actively ignores the mating cells.
Completely. When they stimulated the threat radar, they found that 0 out of 18 recorded mating cells fired a spike. 0. Even though the physical nerve fibers from the threat radar are woven all throughout that general area, they refuse to connect with the mating cells.
It's an act of segregation. The brain is ensuring there's absolutely no crosstalk. Because, I mean, if you think about it, if the threat radar mistakenly triggered the mating cells during a home invasion by a predator, it would be evolutionarily disastrous for the mother and the newborn pups.
The selective wiring prevents those crossed wires. The plasticity is targeted with laser-like precision.
So just to lock this in, the brain's rewiring is highly selective. The PA connects strongly to aggression cells, but actively ignores mating cells, preventing crossed wires during motherhood. Isn't that fascinating how the brain compartmentalizes? Think of how you instantly context switch between your professional self and your parenting self. How well are your own circuits segregated?
It's a really good analogy.
So if we trace our steps, the hardware is fully upgraded. The highways are wide, the engines are revved, the wiring is perfectly segregated to only trigger aggression. But here is the final puzzle piece.
Okay, let's hear it.
If the hardware is fully built out and supercharged, why isn't the mother mouse just constantly aggressive 204/7? Why does she only attack when the pups are actually in danger?
Because having the hardware isn't enough. To understand the day-to-day regulation of this behavior, we have to look at the software. And the software driving this entire system is oxytocin.
Oxytocin. That is completely counterintuitive. It's universally known as the cuddle chemical, the hormone of love, trust, and bonding. You're saying it's the fuel for violence.
It is a paradox, isn't it? But the mechanism is undeniable. Let's go back to our threat radar cells, the PAESR1 cells. When the researchers examined them, they found their surfaces are absolutely covered in oxytocin receptors.
So the threat radar is primed and waiting for oxytocin.
Yeah.
Where is the oxytocin coming from in this context?
When the newborn pups physically suckle on the mother, That sensory input travels up to a brain region called the PVN, the paraventricular hypothalamic nucleus.
The PVN.
Right. The PVN is essentially the brain's main oxytocin factory. It floods the system with oxytocin, which then binds to those receptors on the threat radar.
So the oxytocin acts like a security keycard. The hardware is fully built, the engine is ready, but the threat radar is locked down. It can't send that massive signal down the highway until the oxytocin keycard swipes it open.
That's exactly how it functions. And to prove this, the researchers designed a brilliantly simple test. They called it the separation experiment. They took the pups away from the mother for 24 hours.
What happens when you remove the physical act of suckling?
The oxytocin levels drop and behavioral aggression plummets. But more importantly, they wanted to see what was happening inside the cells. They looked at the brain in real time using calcium imaging. They used a fluorescent indicator called GCAM M6s.
Wait, calcium imaging, how does that work?
When a neuron fires, calcium rushes into the cell, so GCAM T6s actually lights up, allowing them to literally watch the brain firing in real time.
Wait, it literally glows?
Literally glows. And when they imaged the threat radar cells after 24 hours without the pups, those cells were dark. They completely stopped responding to intruders. Without the oxytocin, the radar was turned off.
But then they did the rescue phase of the experiment, which is fascinating. How did they try to turn it back on?
They did two different things. First, the natural way. They just returned the pups, the mother nursed them, the oxytocin flowed, and the aggression instantly returned.
But the second way was artificial, right? They kept the pups completely out of the...
Yes, no pups around at all. Instead, they used lasers to manually stimulate the oxytocin-producing PVN cells in the mother's brain. They artificially swiped the keycard.
And without a single pup in the room, just by releasing that oxytocin, her aggression instantly spiked right back to pre-separation levels. The laser triggered the oxytocin unlocked the radar, and the fierce protector was back.
And to be absolutely certain this was the mechanism, they introduced pharmacology. They injected a drug called L-371,257, which is also known as OXTRA. This drug physically blocks the oxytocin receptors on the threat radar.
It jams the lock.
Exactly. When they jammed the lock, even returning the pups or firing the laser couldn't rescue the aggression. Conversely, when they applied a different drug called TGOT to brain slices, a drug which perfectly mimics oxytocin, they saw the excitability of those radar neurons shoot right back up.
This makes me wonder about the hardware we talked about earlier, the multi-lane highway, the revved-up engines. When the pups are removed for 24 hours and the oxytocin software is gone, does that physical bridge disappear? Does the brain tear down the highway?
That is perhaps the most profound finding of the entire paper. No. The circuit stability is maintained. Even after the pups are removed and the behavioral aggression drops to zero, if you artificially stimulate that PA to VMHvl pathway with optogenetics, the mother will still instantly attack.
So the highway's still there. The race car engine is still there. The structural rewiring of the synapses is fully intact. It just lacks the oxytocin software to run.
Precisely. The oxytocin is acting as a real-time live feed of the pup's status. Suckling equals pups are present and vulnerable. It provides the daily authorization to use the upgraded hardware. It ensures the mother doesn't waste vital energy or risk injury getting into fights when her babies aren't even around to be protected.
Let's do our final summary of this amazing system. Oxytocin acts as a real-time, need-based volume knob for aggression. Cup suckling provides a live feed that authorizes the brain to use its upgraded fighting hardware. Think about that in your own life. What is the oxytocin in your own daily life? What external trigger gives you the sudden authorization to tap into a higher level of focus or energy?
It's an incredibly elegant, fail-proof system of biology.
If we synthesize this whole biological algorithm we've mapped out today, pregnancy and the act of giving birth cause a physical, structural upgrade to the brain's hardware. The VMHvl action center gets highly excitable, and the synaptic bridges from the PA threat radar become massive and strong.
But that upgraded hardware remains dormant without authorization. It requires the software oxytocin, which is only provided by the live feed of pups suckling. The oxytocin acts as a real-time security clearance, guaranteeing the mother only engages in dangerous fights when her vulnerable pups are actually present and need her.
From the initial mama bear instinct we talked about at the start, down to the exact microscopic proteins, electrical currents, and chemical keycards making it happen. It's remarkable how a behavior so primal is regulated by such a sophisticated microcircuit.
It really is. And if we connect this to the broader implications of neuroscience, it leaves us with a truly provocative thought. We just established that the physical structural rewiring of the aggression circuit remains fully intact in the brain even after the pups are temporarily removed.
Right.
It just sits there, completely invisible, waiting for the chemical key to turn it back on. It implies that mammalian brains can hold ghost circuits.
Ghost circuits. Networks that are completely built, fully functional, but entirely silent until the environment provides the right chemical trigger.
Exactly, which makes you wonder, what other fully formed specialized neural networks are lying dormant inside your own brain right now? What ghost circuits are completely invisible, just waiting for the exact right environmental cue or the right chemical trigger to suddenly change who you are?
Think about that the next time you feel that protective instinct rise up out of nowhere. Thanks for taking this deep dive with us.